So today I will just give you an overview about what we are doing in Sheffield in advanced therapies, in particular gene therapy, and I will give background information. I understand that the audience is with multidisciplinary audience, so I will give some background information about gene therapy, advanced therapy, and I am happy to answer any questions at the end. So we use viral vectors to develop gene therapeutics for neurodegenerative diseases, in particular disorders that affect the motor system. So this is just a disclosure slide, no conflict of interest, but I would like to list a couple of tasks that I am doing in different committees, boards, and scientific advisory boards, and etc. So I will start with this, advanced therapies. So this is a term used to cover highly specialized medical treatments, such as cell and gene therapy. And I will focus today about gene therapy, and I will not talk about cell therapy, basically. It's a topic that is advancing very well, and the sector is grow sharply in the last few years, with the expectation to reach 14 to 21 billion per year globally by 2025. And gene therapy uses viral vectors in most of the cases. It's not restricted to viral vectors, but most of the applications use viral vectors to, and these viruses have been modified to remove all the harmful properties to transfer gene therapeutics into patients. So gene therapy made a very successful comeback after some difficult time in the early 90s. And that was mainly because of some investigators were cutting some corners in terms of safety, but it has been a very, very good time for gene therapy in the last 10, 20 years. And many pharma companies came back, and you can see it from the number of market authorizations for products based on viral vectors in the market now. And it's the expectation that will increase. And this is based on discussions that I had with representatives from major regulatory bodies like the FDA or the European medicine agencies. So viral vectors, viruses are used as carriers, and the therapeutic gene is transferred into cells using these carriers, which have been modified to remove all the harmful properties. And the most attractive viral vectors that is being used at the moment, mainly for the applications in the brain and spinal cord, is Adeno-Associated Virus. And I will tell you why in a few minutes. Adeno-Associated Virus, you can see in this slide basically that we don't use wild-type virus. Instead, we take most of the viral genes and proteins out to make space, where we insert the therapeutic gene, and we use separate plasmids to engineer a virus in the lab. And most of the sort of historical data here, so adeno-associated virus exists in different serotypes based on the capsid of the virus. And if you see here on the left side, pre-2002, only a few serotypes existed or were identified at that stage. But post-2002, you can see the large number of serotypes that were generated or identified. And these serotypes exist, most of them in natural serotypes. And AAV adeno-associated virus is very common in nature, and probably in this audience, I would say 50% or more of us, we are seropositive for one of these serotypes, because AAV it's not known to cause any human pathology. But some of these serotypes are engineered in rational design in the lab and to increase their tropism, for example. And they are widely used in clinical trials, as you can see in this slide. This slide summarizes different route of administration of these viruses to treat, for example, eye disease or brain systemic delivery to treat hemophilia, for example, and some of the good results were reported for hemophilia trials recently. In Sheffield, we have a large pipeline at Citron for gene therapeutics based mainly on AAV, and developing a GMP manufacturing facility to generate clinical vectors to treat patients, training and skills, MSc course for advanced therapy, commercialization. We have startup company in place based on gene therapeutics for some of our products, biobank for patients dosed with advanced therapies, and of course, all this linked to very good and excellent expertise in clinical trials, and these are some examples. So again, this slide summarizes some of our programs. I will talk mainly on two today: the gene replacement for spinal muscular atrophy and gene silencing approach for motor neuron disease or amyotrophic lateral sclerosis. So we use mainly Adeno-Associated Virus serotype 9, and the reason for that, because this is a gold standard for CNS applications, brain and spinal cord, for the simple reason because if administered systemically in the blood, it can cross BBB, and this was reported both in animals but also in spinal muscular atrophy patients. And you can also administer it directly intracerebrospinal fluid to target the brain and the spinal cord. And as I said, I will focus mainly on the first program, which is spinal muscular atrophy and SMN replacement. And spinal muscular atrophy, this is just a background slide showing that this is quite a severe disease in most of the cases. 50% of affected children die before the age of 2 years, and it's caused by deficiency of the survival motor neuron SMN protein. And basically, this leads to significant reduction of the SMN protein and loss of function and motor neuron degeneration in the spinal cord. And most of these babies or kids die because of the respiratory failure. So we designed this program to use AAV9 to restore the level of SMN proteins. And as you can see here in this slide, we had a program that progressed very nicely from discovery all the way to orphan drug designation and MHRA regulatory and toxicology regulatory studies, and we were about to submit for approval at this stage. So this is a proof of concept in the animal model showing that when you treat these mice at 1 day post birth, you can increase survival of these mice compared to the non-treated mice or treated with a virus control, which they die on average 11 days after birth. And we published this proof of concept in a prestigious journal, Science Translational Medicine, in parallel the same summer, basically in 2010, with a group in Ohio State University, and we secured funding to progress it to clinical trial. But the group in the US went faster than us because they had a GMP manufacturing facility in-house and generated this virus to treat these kids with exactly the same approach. And you can see that they reported the fantastic Avex's clinical trial, they reported some excellent, remarkable outcome in these kids affected by SMA. And most of them, you can see here, based on the historical study, at this level, 20 months of age, only 8% of the kids or babies are event-free. Basically, they have no respiratory failure. So when these kids were treated, you can see that they survived very well and had no event at all. Most of all of these patients, which was a very remarkable outcome of gene therapy in this case. So I will now move very quickly to a second program that we are doing, which is gene silencing for spinal for ALS, amyotrophic lateral sclerosis. So just briefly, ALS is it's quite one of the devastating neurological disorders affecting adults. And it's an orphan condition, rare disease, but it's one of the third common neurodegenerative disorder. You can see here it's basically affects male and female, and it's caused by motor neuron degeneration in the cortex and the spinal cord, leading to respiratory failure, and most of patients die within 2 to 5 years after the first symptoms. And this is a slide just summarizing where the defects and degeneration occur. So you can see here in the cortex, this layer of motor neurons degenerate, but also in the spinal cord, ventral horn of the spinal cord motor neurons. This is healthy motor neurons, obviously, but in patients, these motor neurons degenerate. So in terms of genetics of ALS, the first gene that was discovered is the superoxide dismutase 1, SOD1, and this was discovered back early in early 90s, and the first animal model was generated based on this animal model, and it allowed the field to progress, and most of the therapeutic approaches were performed in this animal model. So we designed one of our strategies for gene silencing, SOD1 gene silencing for familial ALS, and the reason why we choose gene silencing in this case, because mutations in this gene lead to motor neuron degeneration and ALS by a gain of function, a dominant effect and a gain of function. So silencing and a reduction of the mutant form of SOD1, our hypothesis is that will lead to improvement of the phenotype. And indeed, so the first proof of concept that we generated was using a lentiviral vector. It's another virus. And the discovery that we did was that when we used the lentiviral vector, pseudotyped with the rabies G envelope. Just to confirm, this is not a rabies virus. This is a rabies G protein, glycoprotein. And when we deliver this lentiviral vector in muscle, it can be taken by nerve terminals of motor neurons, retrogradally transported to the cell body located in the ventral horn, and express the therapeutic gene. And indeed, you can see it here using beta-gal as a reporter gene to label, really to transduce motor neurons specifically in the ventral horn. And using this approach, we designed short hairpin RNA to target human SOD1, not mouse SOD1, because these mice, transgenic mice, they express a human mutated form of SOD1, and this mutation exists in human patients as well. So we taking cortical neurons from these mice, they express the mutated form of SOD1, and when we infect these motor neurons with lentiviral vector that express shRNA targeting a human SOD1, you can see that we reduce the level of the protein, but also the level of mRNA in this in vitro model. And then we took this strategy a step further in the animal model. We treated these mice at different level of muscles, legs, intercostal, diaphragm, and also facial muscles, because all these muscles are affected in ALS patients. And you can see here that basically the outcome of this treatment in mice that, if left without treatment, they develop the disease on average around 100 days, and they die on average 130, 140 days after birth. But when we treat them, there is a significant delay in the onset of disease, but also a remarkable extension of the survival of these mice. And this image is just to confirm that the shRNA delivered by lentiviral vector that can be expressed in motor neurons in the spinal cord by colocalizing the reporter gene with a marker for motor neurons of the spinal cord. So obviously we wanted to take this to the clinic, but the only hurdle that we had was that the manufacturing of lentiviral vector to target all the muscles in human, it's just too expensive to manufacture the clinical grade vector to target all the muscle. That's why we looked at alternative, and Adeno-Associated Virus serotype 9 was a good alternative based on the promising results that we had in spinal muscular atrophy, both the animal model but also patients. So the team, both Tom and Joe in my team, generated an AAV virus expressing a short helping RNA, but instead of delivering in muscle, they delivered this virus into cisterna magna, in the CSF, cerebrospinal fluid. And this virus can be taken by this fluid all the way in different part of the brain, but also down the spinal cord. And indeed, this images confirm that. You can see different part of the brain here, and in the second image in the middle, but also in the spinal cord. So they treated the SOD1 transgenic mice, the animal model of ILS, at postnatal day 1, but also postnatal day 40, 2 time points, so different groups, not the same group, obviously, and followed these animals at different stages using different motor assessment and histology at the end of the study. So this is just to confirm again why we choose cisterna magna as an efficient route of administration delivery of AAV9. And again here, showing nicely, targeting different part of the brain and spinal cord. So when we followed these animals at different stage, you can see that the treated animals in red don't, this is the neuroscoring, and neuroscoring parameters are listed here: onset, abnormal gait, dragging one limb, etc. You can see that the treated animals in red here, they don't develop the disease. It's only after 110 days of age when we see some animals developing some symptoms, while the animals treated with the virus control, you can see that they develop the disease even at this stage around 40 days of age. So the onset of disease obviously was delayed in red here, the treated animals compared to blue. The same thing for other parameters like rotaral test, when we place animals on rotating rod and we measure the latency, the time to fall, when they fall from the rotating rod. And clearly you can see a clear difference, significant difference between the treated animals when we deplete the mutated form of SOD1 compared to control mice. The survival of these animals also was delayed. You can see here a clear difference between treated animals versus controls. So the effect was not only basically on the behavior, but also you can see the number of motor neurons. It was very nicely delayed or protected in using different markers here in treated animals compared to controls. We looked also at astroglia and microglia activation, reactive gliosis, and clearly after treatment with shRNA against SOD1, we reduced reactive gliosis in the brain and spinal cord. One of the good outcomes from this study was to design a biomarker that can be used in a human clinical trial. So we took cerebrospinal fluid CSF by lumbar puncture, so at the level of the lumbar spinal cord, and we measured the level of SOD1, human SOD1 by ELISA. And you can see clearly that after treatment of these mice by shRNA against SOD1, we deplete the level of SOD1 in the CSF compared to control, and we obtained around 63% depletion of SOD1. Moving forward, this was a key element for the regulatory in terms of safety and specificity of our sequence of shRNA to deplete SOD1, and we had to do off-target effects study in 2 cell lines, first in HEC293 T cells, but also in astrocytes. These are astrocytes derived from iPSC human cells. And clearly, these studies showed that the specificity of our shRNA. And this is critical moving forward to clinical applications of this approach. So to summarize, this is a great proof of concept in terms of using AAV9 to deplete SOD1, leading to motor improvement, a biomarker showing that we can deplete SOD1 in CSF and can be used safely into human clinical trials. And we are talking now with a company in the US to take this approach to clinical applications. But the good news is, based on this concept of SOD1 depletion, and in collaboration with Biogen, our clinical team in Sheffield, undertook this clinical trial by delivering antisense, oligoantisense at the lumbar level, intrathecal, and followed this SOD1 ILS patients with SOD1 mutations. And clearly, this treatment showed that a very, very significant beneficial effect in ILS patients. And this is reported in New England Journal of Medicine, which currently in PRIS, and it will come out shortly. I would like to thank the team that they did the job, the work that I described today, and in particular the members of the team highlighted in blue here, but also the collaborators highlighted in blue as well. And finally, the funding bodies that supported us to do this work. Thank you very much. And this is just my contact data in case if you would like to get in touch. But if you enter my web page, you can get my email address, but also my PI contact detail. Thank you.